Dr. Shane Wright, who is going to talk to us tonight. There's his topic. And he's a senior lecturer at Auckland University with, he's a leading evolutionary biologist involved in global research in plant evolution. And he's spoken to us before about Metrosideros, the evolution and dispersal of that. So our Rata and Waitakere spreading to New Caledonia and points north right up to the Hawaiian Islands. You might say it was our first successful export. And then more recently, though, he's actually solved the riddle of why tropical areas host vastly greater numbers of species per area than in higher latitude areas. And using DNR and PCR tools, he's able to show that the plants near the equator have a higher metabolic rate, which leads to more genetic variation, and that can be passed on to subsequent generations. So plants evolution is very much part of his interest, the major part, but tonight he's going to be talking about something one step up from plants. I'm going to ask Shane to come forward now and tell us about moa in Aotearoa.
Thank you. Kia ora tātou. First of all, can we just turn the lights down? I don't like speaking as if I'm in a fishbowl. Would you mind switching them both off? Is that okay? Yeah. All right, so part of the research that I've been doing recently is on this wonderful group of birds that have only recently departed, dearly departed. I'll just wait for everyone to come. Take a seat. So moa have only been extinct for 700 years. That's a mere heartbeat in terms of evolutionary time, and their ecological presence still resonates in the country, even though they're now very much missing. This is one of the 8 or 9 species. I say 8, other people say 9. And this is the crested moa, which was endemic to Westland. So it was the moa that was able to deal with high rainfall. And the fact that we had 8 or 9 species crammed into this tiny space, which is Aotearoa, New Zealand, was rather exceptional in the sense that the ratites, which are the same group as our moa in Madagascar, there were only 4 species. So how tiny little New Zealand, half the size of Madagascar, managed to have 8 moa species is anyone's guess. Some people, as I said, say no. So the lecture this evening is about the modern understanding of moa. There has been a great deal of gained knowledge in the last 15 to 20 years, and I'm going to be focusing on much of that as well as my own research. So what are these beasts? So we know they're birds. There is a ratite, it's a cassowary, still living, fortunately for the Australians, not for us. The class is aves, the birds, but these things are infraclass or superorder Palaeognaths. Now, what does that mean? Paleo means old, and gnathy means mouth. So somehow they've been given the name old mouth. And part, again, of what I'll be talking about tonight is to explain that. Now, the ratites, which are the survivors of Palaeognaths, including our moa, as I said, only recently extinct, do come from flighted birds. Here is their ancestor, which is the Thornus in the late Cretaceous. The Cretaceous is the period just before the extinction of the dinosaurs. And The Thornus is also a Palaeognath, is also something with an old mouth, in the way that the ratites have an old mouth. Okay, I'm taking a bit of a risk here in the sense that I could bore you all senseless. But I do want to say why moa are so special, so different. And it relates to this structure here. So what we're looking at is the skull, and we're looking at the skull lying flat with the palate upward. Now the palate, you know, is just the roof of your own mouth. So the palate in the Palaeognaths, the old mouth, still has this big blue thing here called the voma, and sequence of bones that are all fused together. Now, that is the state that the bird mouth was in during the age of dinosaurs, and it links the birds to the reptiles. The reptiles have this structure. So moa are Palaeognaths still with this structure. Now, the voma, which is the bit in the middle, has done something rather exceptional as we step from Palaeognaths into the modern birds. When I say modern birds, I mean the sisters to the moa. In other words, all the other birds that we see, chickens, ducks, sparrows, kōkako, doesn't matter what it is, they are in this form. Now what's happened? First of all, notice that the bone, the blue bit, the blue bone, has become long and thin. It is getting itself out of the way to allow for the modern birds to make a rather marvelous step in terms of their adaptation, because they have reduced the bone to allow the upper bill to be free to lift. Now a moa can't do that, a kiwi can't do that. These are ratites, Palaeognaths. The upper jaw is frozen to the skull in this structure here. So the voma has shrunk, and the pterygoid and the palatine have done something also rather special, which is that they have produced a little articulation between the green and the yellow, which then allows the bill to rise and fall. It would be as if your own palate, when you opened your mouth, you dropped the mandible to take some food in. The top, the maxilla, also rose. The modern birds can do that, we can't. And nor could moa, and nor can kiwi. So let's just take the next step and look at this new mouth, the neonath. Neo means new, nath means spell. So the neonath has a mobile palate. And that is all the birds we know in the great radiation of birds, with the exception of the ratites. So the ratites, which are, you know, our kiwi, moa, the elephant bird, ostrich, so on and so forth, these are the archaic ones that still have a Palaeognath mouth. So you can see that we have the colour there, which is purple and teal and red. So at the point where the red and the teal are making contact, you have a little point of articulation which allows that bill, the maxilla, the top of the mouth, to rise as the mouth is opened. Now, why? We don't know, but I'll take a guess. The birds have lost their teeth, and there was a very good reason to do that, because the bird head would otherwise be too heavy, because teeth are very heavy parts of a mammal, or indeed of the archaic birds, and also of reptiles. So they've lost their teeth probably as a weight-saving device. And in doing that, you then have the challenge or the problem that you have to swallow things whole. And to do that, you probably need to increase the gap to the maximum that you possibly can, means being able to lift the maxilla at the same time as you drop the lower jaw, the mandible. That's my guess, I don't know. But we, with the moa and the kiwi, were holding parts of bird evolution that dated back to the Cretaceous. These very old ones that would still not lift as they were dropping, lift the upper jaw as they were dropping the lower jaw. So a mobile palate goes with a transitional life form, what are often called the living fossils. A mobile palate is, as I said, all of the rest of the birds, the bottom ones that we know so well. And there it is working. You know, when you are fighting with a seagull for your fish and chips, you can often be taken by surprise about how much they can get into their mouths. I also had this experience fishing off Stewart Island, where we were with, you know, with these very able fishermen on a fishing boat, and they were throwing these great pieces of fish overboard, including the frames and all sorts of things. And the albatross that were waiting for this to happen were managing to open their mouths to these enormous widths to get the fish in. So this hinged upper jaw, lifting the maxilla at the same time as you are dropping the mandible, is a characteristic of the modern birds, which dear old kiwi can't do. So the kiwi still has a jaw that is essentially reptilian, or at least a maxilla that is essentially reptilian in that frozen state just prior to the evolution of this new form. It has a fancy name, which is cranial kinesis. Spare me, please. But cranial kinesis just refers to the mobility of the upper and the lower jaws. And cranial kinesis belongs to the neonates, which is basically all of the birds, the modern birds, other than the ratites, including mole. So with the palate frozen, you're down here with Palaeognaths, which is called an infraclass. It's inside the birds, but they are so profoundly different from the neonatus birds that we don't call them a different order, we call them a different infraclass inside of them. And so these old ones, and here they all are, we call them Gondwanan. Their distribution, at least in the last 80 million years, has been in the southern hemisphere on the lands of Gondwanan, Africa, South America, Australia, New Zealand, and so on. We call them Gondwanan. Their origin, see the faunus. You saw that flighted Palaeognaths in the first or the second image that I showed you. So there is a flighted bird that is the ancestor to the radiation of the moa, or at least the ratites, including the moa. That is all happening at the point coming through to that red line, which is the moment of extinction of the dinosaurs, the Katie boundary. And you can see they're radiating just prior to that, in other words, diversifying into different groups, and then they come through on the different land masses, the ostrich here in Africa, Dromaeus and Apteryx and so on, or at least Apteryx, the kiwi in New Zealand, Dinornis in New Zealand, Megalopteryx, Monomopteryx, and so on and so forth. So we have this little group of birds which in our minds we call flightless, but their origin is actually flighted. Because if you came to Earth in the late Cretaceous, there were no neonates. They had not yet evolved. It was the Palaeognaths that are flighted that are doing the business of occupying the niches that birds now occupy today as neonates. So that's a long way of trying to say why these wonderful birds that we've just lost in the last 700 years are so special. And it's so unfortunate that we are missing them, because they're the equivalent of the tuatara, in the sense that the tuatara is also a missing link. All these birds are missing links. Now, moa here in New Zealand, Aotearoa New Zealand, were rather different in so many ways, quite apart from the fact that they are they are what we call slow K-selected. These are organisms that are adapted to living in landscapes and communities that are full, that are packed with life and packed with competition. They also, as we'll see later in this talk, they were also eating some of the worst food that you can imagine a bird would choose, bits of stick almost. That seems to have slowed their rate of growth, so much so that unlike all of the modern birds again, the bones are actually moving slowly enough in their growth to have growth rates. Some seasons are more productive, other seasons are less productive, winter and summer, and so the bones were moving in their diameter growth with growth rings as if they were tree rings. This is very unusual for birds, and it's thought from the nature of these growth rings that actually moa, unlike modern birds, which develop into adults within weeks or months, unlike the modern birds, they were taking up to 10 years to mature from the juvenile form. So that's very similar to being. These were birds that were, so profoundly K-selected that they were behaving as if they were a human child in terms of their development. They were doing something else very, very different. So there's a girl, and there's a boy. The sexual dimorphism between male and female in moa was so profoundly large, the difference was so profoundly large, that these initially were given different species names. And it was only in the early 2000s when it was discovered, hey, there's an XX chromosome in this one, and there's an XY chromosome in this one, that it was suddenly realised that these were part of the same species, and in fact the same populations. And the male was relatively tiny and the female was huge. Just so that I'm not boring everyone just with the sound of my voice, why? What do you think? Why do you think in a case-selected organism like this that is struggling with competition, there are huge numbers of individuals, high density in the population, there's a great struggle to live and to reproduce? Why would you do that? Yes. Brilliant. It's a hurry effect. So exactly, and if you're going to petition the niche and the female is the egg bearer, I think it would be a good idea to get the biggest egg you can. If you're producing just a very few young, and those young have to come out to be competitive immediately. So it's a waste of time having large males, all they do is fight. Why don't we concentrate most of the energy that's finding that community through the female population, large females? Again, we don't. And this is just a guess. But this is so unusual. The other ratites do not do this. Even the elephant birds of Madagascar did not do this. This is, again, a uniquely New Zealand form. It also seems that there were more females in the populations. All that matters here is the pie chart in the centre. One's from Bell Hill, which is somewhere the eastern South Island, the Turritin Valley, somewhere else in the eastern South Island. And this is the total population, and the black component is the female part of that population. So females don't just seem to have been big, but there seem to have been more of them in these functional populations. And especially the situation of the Turritin Valley is rather remarkable. Where did they come from? Well, if you just consider the ratites, you get this long diatribe from most biogeographers about the Southern Hemisphere and about Gondwanan. You see Gondwanan, there's a whole unit there, and then it's fragmenting. Africa's departing first, which is the purple bit. Notice that New Zealand is departing here at about 80 million years ago. That's Zealandia. And South America, red is becoming detached during the early part of the age of mammals, and Australia is still stuck to Antarctica right through about 40 years ago. But when you ask of the old flighted Palaeognaths, where were you? Where are your fossils? They were actually worldwide. So it's not the Palaeognaths that are Gondwanan. It seems to be this radiation of flightless Palaeognaths, which are Gondwanan, that came after the fullness and which have evolved, all of them, to be these large brown-looking birds. So we have ostrich here, we have the elephant bird here in Madagascar. We have more in kiwi green in Zealandia, so many of them. Asian line moa, 3 kiwi. We have the blue for Australia, which is the cassowary and the emu. We have the rhea in South America. Oh, we have a flighted Palaeognath surviving into the bottom earth. What's that? Tenement. Fantastic. Thank you so much. So that on the left is the nice sequence that we should see if the breakaway sequence of the continents, Africa going through first, so this is basal, coming through to the more terminal or more derived forms with the final departure of South America and Australia up here. That's what we should see, but unfortunately when we do the gene sequence, that's what we see. Go figure. Why? Who knows? People are coming up with all sorts of funny ideas about things being able to fly and disperse and then re-evolving flightlessness several times, and you know, it gets very messy. We don't know. One thing I will say though is that you have to rely on the consistency of a molecular clock. And if your molecular clock is not behaving itself, and there are many reasons why that can happen. For example, in polar climate, it ticks more slowly. In a smaller population, it ticks more slowly. If that's the case, then you can get a mess on your hands if you're trying to look into deep time and build a phylogeny just from DNA. I prefer morphology to be able to spell, but we can't see that far back. I'm not going to try and explain why that's different otherwise, though. But we do know more have been here, at least from the fossil record, for at least 15 to 20 million years, because there at St. Bathans in Otago, in the old lake bed, we have a moa eggshell, or at least a piece of a moa eggshell that is Miocene, somewhere between 50 and 20 million years old.
Can you point out which is the eggshell, please?
This is 1 layer, 2 layers, 3 layers of the eggshell.
Oh,
yeah. Take my word for it. I mean, I don't know, but that's as it's been presented. Okay. So that is apparently a moa eggshell. Yeah. So you know, we at least have residents for moa here for the last 15 to 20 million years. And then there's all kinds of debate about they may have flown here or they may have been resident throughout, you know, the period of Zealandia's isolation, which is something between 70 and 80 million years from its departure from Gondwanan. We don't know. I prefer the idea that they've been here forever. Where did they live? Well, just before we go to the whole country, here's our neck of the woods. And each one of those little black dots is at least a single subfossil, and sometimes more, and including eggs as well. So in our region, they were everywhere. Sadly, I don't see anything in this field, but they were ubiquitous. They were living right throughout. But they weren't evenly distributed. And this excellent piece of work, again, very new, 2019, by Waitakere, is showing us a couple of things. There's two ways that you can measure presence. One is with species diversity, the number of species. That's here on the left. The other is how many sheep in the paddock. In other words, density. How many individuals per unit area. And that's shown there on the right. Now notice that in the lower eastern South Island, so the darker red colours are more. We're up to 7 species here in the drier parts of Otago coming through into the South. And note that no one wanted to be in Westland. It's peeing with rain all the time. We had one specialist more in Westland, which was Palaeognaths australis, which you saw as the crested moa at the beginning. But moa tended to be concentrated in the lower South Island, and in particular in the eastern South Island, up against the foothills of the Southern Alps, at least in terms of numbers of species. We'll ask the same question of density, and that's the number of individuals per unit space, and in this case we've got individuals per square kilometre. Now notice that the densities are pathetic. They are somewhere around 7 as our maximum in a whole square kilometre. How many sheep can a farmer get into a square kilometre when we've got 64 million sheep in the country? In fact, though, moa were in rather vanishingly small densities everywhere, so that the highest density you might find is maybe 6 or 7 birds in a whole square kilometre. That's 3 pairs in a square kilometre. So this is nothing like what we could do with sheep. So the total moa population was only in the high hundreds of thousands, maybe going through to about 2 million is the range of the numbers for the whole country. But again, note that density is highest here. We also have high density throughout the Eastern South Island. We also have a high density here in the lower southeast of the North Island. The grey is the dry places. So I don't want to, when I go hiking, I don't want to go to Westland. I'm going to get hypothermia down there. So they seem to have concentrated both species diversity and relative abundance, the density, where there was least rainfall. Now again, just so I'm not doing all the talking, anyone want to speculate about why it might be this? Not really, because we've discussed this.
When you talking about why the South Island's better than the North?
No, I'm looking at why the east is favoured over the west. Yeah, so what it's drier here. This is where all the moa are in terms of species and density in dry eastern South Island and in the dry lower southeast of the North Island. Why would that be?
People trying to hatch an egg for when it's raining.
So it could be the trees are lower and they can run every.
Fantastic. Yeah, I'm about to retire. You should be doing my job. That's fantastic. So, you know, you can imagine that the reason that the recent pioneering stage of putting agriculture into New Zealand, the first thing you have to do is take out the trees, because what the trees are doing is they're parking productivity above where a sheep can access it. So you take away all the trees, and you then have your productivity in a foot above the ground at the maximum. So the other way of reducing cover, in other words, the size, the height of the forest, and its basic area, which is just the water, is to dry it out. And that will reduce a canopy to a height that's accessible to an animal which is not flighted and which is only able to harvest up to about 3 meters above ground level. Again, I'm speculating. We don't know. Thank you for helping us speculate. But it is interesting that both species diversity and density were highest where forest was actually having the greatest difficulty growing. What did they eat? Well, I have to say, I have due respect for them. You can see that we've got a wheel of potential genera for consumption by moa, and we have wonderful information coming from Jamie Wood and others. This is 2020, where you take the coprolites, in other words, the poos that moa have left in a dry place, and you gene sequence the poos to find out what the genetic signatures for various plants are in the coprolites that have been dropped and survived in the dry space for many hundreds of years. Now, we've got ferns and allies, we've got monocots, we've got climbers, dicot herbs, and so on, but look at the portion of trees and shrubs. So this is exceptional for birds. They're eating bits of lignified tissue, the worst forage you can imagine. Birds eat insects, the modern neonates eat insects, they eat fish, they eat, at worst, they're frugivores. In other words, they eat fruit. But to actually be eating the largest component of their diet as trees and shrubs was rather remarkable. And moa had evolved into that space. And so now what you're looking at is, I've stopped raving on about the palate, and now what you're looking at is the mandible, your lower jaw. So note that we have yellow down here for Australia, and we have a mandible for an emu and a mandible for a cassowary. Now note how gracile, in other words, how fragile they are. And then we have alpine moa, bush moa, giant moa, crested moa, and so on, all the way up to coastal moa. And these mandibles, in fact, the entire skull, were these great clamping systems that could bear enormous stress. So they're not in any way similar to the surviving ratites. The surviving ratites, including the rhea and the ostrich and even the cassowary, are pickers and gleaners. They're picking little bits and pieces, seeds and so on, up off the ground. The moa was, dealing with woody plants. and breaking them up and consuming them. These are just, so the reason you have all of these columns, which is confusing, is that these are the stresses produced by different actions of pulling and clamping and so on and so forth. But the take-home message is the molar mandible and the palate were designed to take enormous stresses. And so they could do that. Those are bits of stick, and they have been clipped by a molar. We know that because this is how molar built their nests. So these were fragments with the eggshell in nests in Otago and Wright overhangs. So you can see that the action of the bill is at once crushing and clipping at the same time to produce that kind of toothbrush form in the short segmented pieces of wood that were then dropped into the nest. They were having to grind this stuff up if they swallowed it, and they were doing that by doing something that a bird should never do, which is filling themselves with rocks. Gizzard stones. So the gizzard stones often lie in a little pile in the middle of the skeleton. They were, because of course, my black teeth. They lost those with the thorns. Can I have them back, please? I need them right now. Sorry, gone. So, my interest, because I'm really interested in botany, but my interest is in actually putting the two together in the sense of the plants and the animals of primeval Aotearoa, New Zealand. And we have these very unusual and unique plants in the country. They're called Biogeographer. They have high angles branching. They have tiny leaves, and they do something that a plant should never do, which is that they park the leaves deep inside the shaded body of the plant, where they can't effectively photosynthesize, or at least they don't photosynthesize very slowly. So, because of that, this is an extremely rare and unusual habit or life form in plants worldwide. But for some weird reason, 10% of our living plants are developed. They do this. Why? Yes. There are two ideas in hot contest about why this should be the case. One is moorgrass, and the other is climate. The climate argument is that during the Pleistocene, which is the cold ice age that we've just come out of, only 15,000 years ago, it was valuable for a plant to be able to preserve some of its photosynthetic tissue inside a warmer body that would experience less frost and wind, and that is the climate hypothesis. In short, the Mott-Browse hypothesis is, well, if you don't have teeth, you can't just wander in here like a deer and just chomp your way through. You've got to use your bill to push into the plant and pull pieces of the plant off and clip them. Now the eye is very close to the tip of the bill in the molar. And so this is potentially a resistant form that will at least slow the rate of harvest, and make it more difficult for molar to effectively browse. Notice that you can't just pick one giant leaf and go away and munch it. The leaves have been fragmented into tiny units, which increase the challenge further in terms of getting enough energy. Plants, some of the plants are also heteroblastic. What does heteroblastic mean? Hetero means different, and blasti means form. So they're heteroblastic between the juvenile and the adult form. And we all know lancewood, Waitakere. The juvenile there, I don't think I'd ever want to try and be a sword swallower and try and get that down the throat. But the adult leaf looks, you know, if I'm a foliivore, in other words, a leaf eater, the adult leaf looks reasonably accessible. But it's not accessible because it's now above 3 meters above the ground. So the juveniles that are in the browse zone for moa are often very, very different plants from the adult. Now, interestingly, this heteroblastic form does not occur where there were no moa. Where's one of the places there were no moa? The Channel Islands, record. So moa browse is a possible explanation here, especially because the change with height from this to this normally occurs just in the zone where you are at the maximum limit of reach for a browsing rod. So there's the juvenile Waitakere on Chathams. And there's the juvenile Waitakere at Antarctica. So, I don't know. So again, I mentioned Jamie Wood because of this wonderful work that's been done with food plants, where you're taking a coprolite, that's an old poo. It looks like stacked spaghetti or lasagna. And they still remain extant where there is a dry rock overhang right throughout the eastern South Island in particular. It's a little bit wetter up here at the North Island. Now, they're full of information. They have pollen information and they have DNA information. And so if you can associate the coprolite with a species, and you can also do that because the coprolite is telling us from some of the DNA what the moa species was that dropped it, then you've got a combination of the moa species and its food. And oh boy, we can now do some, or at least Jamie Wood can. What do you mean we? Jamie Wood can. We can now do some rather nice things in terms of why are there 8 species? I mean, that's ridiculous. There's too many. There's only 4 in Madagascar, which is twice the size, twice the area of Aotearoa, New Zealand. Well, we can associate each of these 4 species, Anomalopteryx green, Gondwanan yellow, Palaeognaths red, and Megalopteryx blue. We can associate each of these 4 species with the food that was in its coprolite on the day that it dropped the coprolite. And if you do that with enough samples, what we are getting for Anomalopteryx is that the thing was living in the forest and it was full of forest plants. What we are getting with Palaeognaths here in red is that it is full of plants that live in the herb field and grassland, so it's living in open ground. For Gondwanan, which is the largest of them all, called the giant moa, Gondwanan robustus. Where are you? I'm neither forest nor open ground. I'm living in the ecotone between the contact point between those two habitats. And these plants are what I have in my stomach, which are the indicators for their ecological address, as it were. So Gondwanan, the giant moa, was ecotonal. It was feeding in this contact point between a low subalpine shrubbery and the forest edge itself, maybe even a Vegas of southern beach. Palaeognaths, with the huge feet, was open ground, feeding on small plants. Anomalopteryx, the bush moa, was in the bush, feeding on all of these weird, variegating understory shrubs that we see in places like Antarctica, the divaricate plants. So what's the blue one doing? Eglapteris. These are specialists. It's a generalist. But it's generalist in the feeding niche, but it's not a generalist in the spatial niche. This is the alpine. So it was stuck in a living space above all the others. Probably because it wasn't being forced to compete, it was eating right across this range of available foods. So I've been studying this just prior to retirement. I have to say I've been disappointed by the fact that there has been this ongoing debate about the climatic hypothesis, or the Morrow-Browse hypothesis that has remained unresolved right throughout my life. I have been doing all sorts of other things with my research, but latterly now I've come to looking at more. And so the first and obvious thing to ask, which someone I should have done 30 years ago, but anyway, is these divaricate plants, just are they mechanically resistant? No one had even done that. So a student of mine, Jarden Howard, a few years back now, 5 years ago, took the stems of the divaricates and their nearest non-divaricate relative. So you have a pair that are basically phylogenetically close, in other words, they're the same but they have completely different life forms. One's leafy, large stems, and the other one is not equal to the divaricate leaves, and as we discovered, incredibly powerful stems. So we put them in a tensile tester here at engineering, and we pair the contrast, as I've just said. Caprosma, Nursini, Nursini, Myrbechium, Myrbechium. So we are contrasting the closest relatives. We did that many times right across the flora. And what did we find? This. So the non-divaricate plant, which is not showing all of these weird adaptations, so let's say Caprosma repiens, these are the megapascals at failure. In other words, pull it apart and pop, it breaks. Megapascals at failure, there's the mean there is just over half the mean for the divaricate plants. In other words, the ones that were showing these adaptations, which we think may have been based on resisting brown spike mold. So we're getting almost 2 times the tensile strength in the divaricate as we are in the non-divaricate. So I had another student, Fergus Wright. Look at this. Who is this Rastafarian? And what is that? We don't just have funny little leaves and strange branch angles, we have something going on with colour. Now, you can call that mimicry, but if you are trying to match yourself against an inanimate colour background, it's called masquerade. And there's debate about whether it's inanimate or not. But Matai here seems to have a cryptic ecology, but only in the junior. Once Matai has grown above Morrow-Browse height, it goes deep green and gives the fingers to anybody that's coming along below. So I'm not too tall, sorry. So what seemed, or at least the question I had in my mind is the leaves seem to be masquerading as stem. The mati, let's see what's going on with colour matching between the stem and the leaf. This was done by a student at Wright. And we did this for juveniles and adults across the heteroblastic divergence between the juvenile and the adult. And we also then used the homoblast, nearest relative. In other words, it's not heteroblast. It's not doing the switch from one weird life form to a normal adult. It's homoblastic and comparing the two. Now, what did Fergus find? Let's just try one. So, mati and mirror, they are, or at least before some naughty taxonomists ruined our little heavy space, they were congeners. Now, mati is doing something unusual. And so what we did was, with the juvenile, what we're doing here is that we are comparing chromatically the foliage of the juvenile to the stem. So the leaf to the stem. And so it's foliage the background, which is confusing, but all the background means is that what are you masquerading against? You're masquerading against the stem if you are the leaf. Because you're pretending to be a stem, so you look bad and you don't get it. This is why people don't buy mati nurseries. They look appalling as juveniles. So this scale here for chromatic is that as you go this way, they are more similar, the leaf and the stem. As you go this way, the leaf and the stem are completely different. They're normal now. The leaf is green and the stem is brown. That's what it should be. But here it's brown, brown. Now, notice that the juvenile is brown, brown. The adult for mati is green, green. That's the simple way. Does Vera do that? No. I'm not divaricate. I've got another secret, but I am not divaricate. And so the juvenile and the adult do not differ in terms of that chromatic scaling between the leaf and its background. So when you see a mati seedling in the nursery, and I'm always so grateful because there's so many of them because no one else buys them. They see them in the nursery and think it's dead, which is what the mower is supposed to think, right? No good. And of course you plant them and you tend them, because I've lived for so long now, you tend them for 30 years and they emerge as this green goddess after about 20 years and become a wonderful tall deep green tree. But they're not going to do that when they're inside more browse height, or at least that's one explanation of this. So the mati seedling has a masquerade between the leaf and the stem as we quantified it with Fergus's work. So the other way of, I hope I'm probably got you trapped here. Tell me to stop if I'm going too long, but I'm nearly finished. Okay. So the other way we can look at this is just ask the question present, absent. And what's going on in the flora where moa are present and moa are absent? What do you mean moa are absent? They're everywhere, right? They're all through the green. Well, no, they're not on the sub-Antarctic Islands. They're not on the Chathams. They're not on the 3 Kings, never were. They're not on the Cumulonimbs. I know what we'll do. We'll take those plant genera that we know can produce a divaricate form and that we know can disperse. In other words, they can cross over the water to get to the Chathams or somewhere else offshore. So instead of having 2,000 species, which is the whole flora to deal with, that gets us down to just a few. And then when we do this, and we ask the question of where divarication is occurring across that diaspora of geography, we've got this. Again, they've been arguing this point since 1980. No one's done this yet, but we have now. We've actually published this in 2021. So what are you looking at? Absent, no moa. Present, moa. So that's the mainland and the land bridge islands. In other words, Aotea, Hotutu, the islands that are connected during each ice age and had moa on. So let's ask the question of absent, non-divaricate. Well, the black circle, which you can hardly see, is expected, and the coloured circle on the chi-square test is observed. So expected and observed are right on top of each other. We expect roughly 16 species to have evolved as non-divaricates where there are no more, and bigamy, they do. Non-divaricate on the mainland, 79. The observed and expected are right on top of each other. Mainland divaricate, yep. They're divaricating on the mainland. No problem with that. 62 of them. Divaricates. What have you done on the remote islands? We expected to see around 10 of you as endemics on the Chathams and on the Sub-Antarctic Islands. We'll get nothing. So the geographic test is also strongly indicative of some kind of effect related to moa presence absence. So my latest student has just finished his position. What was he doing? Well, again, this should have happened in 1980. You're arguing about whether moa want to eat this or this. Tell me what the nutrient content of those two plants, which are sisters, in other words, they're as closely related as they can be, but the morphological difference is enormous. One is divaricate, one is non-divaricate. Tell me what's going on in terms of the nutrients collected in their bodies. Well, what we found was this. What does this mean? Oh, crikey. Potassium, sodium, sulfur. We had to measure this. This is probably why no one did it. Magnesium, manganese, nitrogen. We've got 12 macro and micronutrients here. The green is the non-divaricate sampling, and the red is the divaricate sampling. Now, note for the stuff that really matters, phosphorus and nitrogen, and for really important things for photosynthesis like manganese, the divaricate plant has actually got something worth hiding. They are where all the nutrients are. Yes. Yeah, possibly. You can argue one-on-one for all of these, but this in toto was for 12 macro and micronutrients, and right across them we were getting this completely unknown and now revealed fact that the divaricates were carrying in their bodies a much higher reward that would seemingly make it worth having to carry around gizzard stones in your belly and having to develop these enormous bills to section these plants. So what we're doing is we're trying to look at the challenge from all angles now in the last couple of years of my pre-retirement period. So I got rather excited with all of this because we're starting to at least from my point of view, reduce things. So thank you for your patience. Some questions? Yeah.
If the